Hello, welcome student. We shall start the new topic, that is plastids. You know that it is found in plant cell, yes, mostly found in plant and euglenoids, example euglena. This is found in plant and also in euglenoids, that is in the euglena. And plastid, you know, it is the found in the plant cell, understand? And because in this the photosynthesis takes place and the food is manufactured or food is formed, that's why it is known as kitchen of the plant. Understand? Plastid contains different types of pigments. Plastid contains different types of pigments. On the basis of pigment, it is divided into, on the basis of pigment, plastid, one is Chloroplast, next one is Chromoplast, and last one is Leucoplast. On the basis of pigments and on the basis of their colors, they are divided into Chloroplast, Chromoplast and Leucoplast. And in other way, we also can say that it is divided into colored and non-colored. This is the non-colored, that is colorless, and these two are colored. Understand? Means chloroplast and chromoplast are the colored, and this is colorless. Now, what type of colors? Chloroplast, it is that is green in color. It is other than green, and this is colorless, that is white. This green in color is due to the pigment chlorophyll. Also, you will find later that chlorophyll is of several types, chlorophyll A, B, C, D, like that. So it contains chlorophyll, and in addition to chlorophyll, that is also contain carotenoids or keratin. That is, chromoplast is also contain fat soluble carotenoids, xanthophyll. It also contains pheophil in the Pheophyceae, rhodophyll in the Rhodophyceae, like that. And that's it. And it is found in, especially in the leaf, and it is in the colored part, that is color of petals, color of fruits, like that. And it is the leucoplast is colorless, and in the root tuber it is found. Understand? So, and this chloroplast, its main function is storage of food. This is the storage of food. On the basis of a storage of food, it is of three types. On the basis of a storage of food, it contains both all the carbohydrates, proteins, and fats, and on that basis, their different names are given. On the first amyloplast. It stores the carbohydrate like starch, etc. That is, the reserved food material is carbohydrate, starch, like that. That is in the example potato. Next one is Plast. It contains protein. Reserved food is protein. Reserved food is protein, as for example in the castor. And the third one is, this is the one, this is the two. Plast. This one. In protein, that is bean, right? Soybeans. Plast, the reserved food materials is fats and oil. The reserved food material is fats and oils, and in that the castor and the other. So castor and the we shall discuss on chloroplast. It is found in mainly in the mesophyll cells of leaf. Mesophyll cells of the leaf, they are found. They are oval, elliptical, discoid, separate. They are found different types of found in that. And if we study the chloroplast under the electron microscopes, we get that it is a double membrane structure. That is one. Outer membrane, and this one is called inner membrane. It's both. And the inside bag, understand, is called stroma. In the stroma, we'll see that different types of enzymes, DNA, ribosomes, like that, they are found. And And except the stroma, in the stroma also some discoid or plate-like structure is found. These are called thylakoids, and there are so many thylakoids, means about 10 to 100 thylakoids. 10 to 100 thylakoids combines to form gramina. So the total structure is gramina. Singular gramina. And this is also that. And these thylakoids and gramina are attached to each other. These are called stroma lamellae. And in the thylakoids, these are the thylakoids, about 10 to 100, and about 40 to 60 or 40 to 60 grana found in each chloroplast. 40 to 60 in let it go. So this is the double membrane, except that in this is circular DNA. The ribosomes, because due to presence of this DNA and the ribosome, that is genetic materials, and for the genetic material it is independent, and for the protein synthesis it is independent. That's why it is called semi-autonomous. That's why it is called like mitochondria has also its own genetic material and ribosome, and so these two structures, plastids and the mitochondria, are semi-autonomous because they are not depend on nucleus for their functions. And one thing that the ribosome is also found in cytoplasm and in the plastid. But there is difference. The cytoplasmic ribosome, it 80S type, and this is of 70S type. So this is the structure of chloroplast. Thylakoids, it contains many units. In the that content called quantazome. That is called quantazome, and this quantazome, it contains chlorophyll, and the chlorophyll trapped light for the photosynthesis. Means the light reaction of the photosynthesis takes place in that part, and the dark reaction in the stroma. So that's this is the about the chloroplast, their double membrane structures, and having thylakoids, the disc-like structures, and this thylakoids contains quantazome in which chlorophylls are present, and that chlorophylls absorb the light. understand, and which is helpful in the photosynthesis. So the photosynthesis takes place in the plastids, and that's why it is known as the kitchen of the plant, due to presence of genetic material and protein ribosome. It is the semi-autonomous, and the ribosome is which is found in the chloroplast is of 70s type, while in the cytoplasm is of 80s type. Now the next one is ribosome. You know that it is one of the most important cell organelles of the plant. It is found in both prokaryotes and eukaryotes, although it is a minute structures, and before the discovery of the electron microscope it was unseen, but in the 1953 the work palette observed it and gave the name ribosome, because it is made up of ribonucleic acid plus protein. It is made up of ribonucleic acid and protein. And in the prokaryotes and eukaryotes, they are found. In that it is of S type, and it is in 80S type. This is S means S Bedbugs unit. So, and this is composed of, you see, it is composed of two parts. One is small and one is large. One is small and next one is large. So it is composed of two units, and in the eukaryotes means the 80S is divided into 60S plus 40S, and in prokaryotes it is divided into 50S plus 30S. So these are the two types of, and this contain the ribonucleic acid and protein, and also it contains the, and it is called the site of protein synthesis. They are found in the cytoplasm, found on cytoplasm plus endoplasmic reticulum. When they found on the endoplasmic reticulum, that endoplasmic reticulum is called rough endoplasmic reticulum. They are found in separate and sometimes in the cluster, means many. In the cluster they will find, that is called polyribosome of some minor polysomes. So that is polyribosomes or ribosomes, they are separate. So this is the protein site of protein synthesis, and the 70S and 60S is the S means S Bedbugs unit, and it measures the density and size of the subunits. Understand? And so that is main importance because it is the site of protein synthesis, and it is found in cytoplasm and as well as in mitochondria and the Golgi bodies, which we have already studied. But there is difference between in the cytoplasmic, it is 8Ts, and in the mitochondria and Golgi bodies, that mitochondria and plastid, it is of 7Ts, and which is found in also prokaryotes.